Each cell in our body contains copies of every gene in our genome. The activities of these genes is what contributes to our traits, causes cells to be specialized, and causes genetic disease. Gene activity, also known as gene expression, is when the mRNA that the gene codes for is produced by the cell. When genes are turned on or become active, they are said to be expressed, and the cell produces mRNA from that gene. For genes that are turned off or not expressed, the cell does not produce mRNA from that gene. Why don't we look at a few examples of gene expression in the human body? A muscle cell that relies on the two proteins, actin and myosin, for contraction will have the genes responsible for these proteins turned on. Because muscle cells have no need for the pigment melanin or the hormone insulin, the genes for those are off. Likewise, a cell in the pancreas of a healthy individual would have the gene for insulin turned on, but not the gene for melanin because it has no use for pigmentation. Scientists can compare gene expression in various cells to better determine their function and how our environment, medications, and other factors can affect that function. Sometimes, internal or external factors can cause genes to mutate, which can change their level of expression. A mutation could cause the gene to turn on or off, increase its activity, a process called upregulation, or decrease its activity, a process called downregulation. In the case of a disease such as cancer, one or more genes experiences a mutation, causing a change in gene expression. This change could cause the cell to divide out of control. As these cells continue to divide, a tumor forms. To find differences in gene expression, scientists compare the amount of mRNA produced by the genes in a healthy cell to the amount of mRNA produced by the genes in an unhealthy cell. Why do they look at mRNA? Because if a gene is being expressed, mRNA is being made by the cell. The more the gene is upregulated, the more mRNA is being made. If the gene is downregulated or not expressed, then little to no mRNA is produced. In these examples, gene 3 would be of interest to researchers because of the difference between its level of expression in a healthy cell and an unhealthy cell. To compare these gene expressions, scientists use a tool called microarray, which you will explore in detail in the next video.